So I think we're going to go ahead and get started, and I'd like to welcome everybody back to Brain 101. This is our fourth of six lectures in our first year medical school course, and thank you all for sticking with us up to this point in time. These have been some great sessions. So just to remind you, this is being hosted by Tammy Lack, who you see here, who is head of strategic development for the Department of Neurosurgery, also executive director of the Neuroscience Innovation Initiative. And then myself, I'm Kevin Lillehei, chair of the Department of Neurosurgery. It's sponsored by the University of Colorado Medical School, but also by the Neuroscience Innovation Initiative. And just real quick, the Neuroscience Innovation Initiative is a program that we started several years ago, and it's basically to bring neuroscience together, not only across the campuses of the University of Colorado, but really across the state. And it's really a precursor to the development of a comprehensive neuroscience institute. It's dedicated to integrating all of the neuroscience work that's going on campus and around the state, to enhance the collaboration of all of our neuroscience researchers, and also to communicate. And the mission really of the NII is to make Colorado the healthiest brain state in the country. So having said that, we have 7 broad programs, as you see on this slide on the left. And today, the first program, Aging Across the Lifespan, Neurodegeneration, is going to be our topic with Dr. Peter Pressman and Dr. Huntington Potter. So I'll say a little bit more about that. As shown here, this is our fourth of 6 lectures. You guys are well on your way to completing the requirements as a first-year medical student in the neurosciences. Congratulations. As shown right down here, we will have a graduation ceremony. It's purely elective, but if you would like to attend it on Saturday, June 1st, please contact Tammy to let her know. It's from 9 to 11, and it's really an opportunity to network with others that have been on this Zoom, and to ask any other additional questions that people may have about the neurosciences, and to get your graduate degree. So I'm going to remind people once again that during the lectures, we want to allow 10 minutes at the end to answer questions. And sometimes that's kind of the most exciting part of answering these questions that people have raised. But as the lectures are going on, use the chat function at the bottom of your Zoom and go ahead and put in your question. Tammy will be monitoring all of these and collating them. And then at the end, she'll choose ones to ask our speakers. But any ones that we don't get to, we've been very good at sending out an email sometime later in the week with answers to those questions. On that same note, Tammy sent out this week an email with all of the recordings of the first 3 sessions. So those are available to everybody. So having said that, I want to go right into introducing our topic tonight, which is Introduction to Neurodegenerative Disorders and Alzheimer's Disease. Our first speaker tonight is going to be Dr. Peter Pressman. Dr. Pressman is an associate professor in neurology who specializes in cognitive and behavioral neurology here at the University of Colorado. He's involved in clinical trials at the University of Colorado Alzheimer's and Cognition Center and sees patients in the memory disorder clinic. Dr. Pressman obtained his MD degree from Oregon Health and Sciences University, did his residency at the Chicago Northwestern Memorial Hospital, and then completed a fellowship at the Memory and Aging Center at the University of Colorado, San Francisco. Dr. Pressman specializes in frontotemporal dementias and primary progressive aphasias, and is particularly interested in verbal and nonverbal communication deficits in neurodegenerative disease. He's going to be talking with us this evening as a brief overview of neurodegenerative disorders, leading into a general overview of Alzheimer's disease. So Dr. Pressman's talk will be followed by Dr. Huntington Potter. Dr. Potter is a professor of neurology and director of both the Alzheimer's Research Program and the Linda Cernick Institute for Down Syndrome and the CU Alzheimer's and Cognition Center. Previously, Dr. Potter studied, researched, and taught for 30 years at Harvard University. He then designed and directed the Florida Alzheimer's Disease Research Center and the Johnny B. Bird Senior Alzheimer's Center and Research Institute. He's currently expanding current therapies that Alzheimer's disease is mechanicalistically related to Down syndrome. We're going to hear a little bit about that tonight. He's author of over 100 scientific articles, books, and patents, has received numerous awards for his work, and is a fellow of the American Association for Advancement of Science and the National Academy of Inventors. Of interest, his electron micrographs of DNA are on permanent exhibit at the National American History Museum of the Smithsonian Institute in Washington, D.C. So having said that, Peter, I'm going to turn this over to you. Let me stop sharing here. You're on mute, Peter.
Thank you, and thank you, everybody. Let me just load things up here if I can. There we go. So thank you again, Dr. Lillehei, for the kind introduction. Peter Pressman, cognitive behavioral neurologist at the University of Colorado. I'm going to be talking tonight about Alzheimer's disease-related disorders. And I'm going to start with some good news because I think, well, we all know the bad news already. And I think we've heard a lot of it. It helps people to keep going both in the clinic and in research to focus on real reasons for hope in this field as well. I'll have to go through some definitions and then discuss how we diagnose neurocognitive disorders and neurodegenerative conditions, how we treat them, how we may prevent them, and give some helpful tips along the way. So, starting with some good news. There's been several publications that suggest that at least in countries such as the United States and many European countries, age-adjusted incidence of dementia, meaning the chances of a 70-year-old getting dementia now, are significantly less than they used to be, say, a couple decades ago. Now, there's more people getting older, so at a population level, we still need to watch for this. Dementia is becoming more common, but all of our individual odds are getting better. Some of this may be due to the fact that we are more able to intervene across the lifespan by paying attention to things like smoking, head injury, getting better educations, making sure that all that good advice doctors should have been sharing for a long time is actually being followed. Now, I need to define a few things here because this is a common source of confusion. When we use the word dementia, this is what we call an umbrella term. So this just means thinking problems getting in the way of somebody's ability to go about their life independently. There are many different causes of dementia. Somewhere I once read over 300 different causes. Many of those are what we call neurodegenerative, and we'll come to that. Those would include things like Alzheimer's disease, frontotemporal dementias, Lewy body dementia, and other related disorders like Parkinson's disease. However, other reversible things can also contribute to a dementia. Things like using too much alcohol, certain toxins, or just metabolic conditions, endocrine or hormone problems, and more. So this is one of the reasons why we emphasize that if you're having thinking problems, you shouldn't just shrug and say, well, there's nothing that can be done. In fact, it is important to get that evaluation because there is a reasonable chance that this is something that can be slowed and sometimes stopped or reversed. Now, when we say neurodegeneration, those are the situations where, as of yet, we don't have a good way to reverse. So these are conditions like Alzheimer's disease that continue to progress over time, where we see a loss of brain tissue or nervous tissue, and parts of the brain shrink faster than we might expect as just part of normal aging. So classic examples beyond Alzheimer's disease would include Parkinson's disease, which impacts movement more, or motor neuron diseases like ALS, also called Lou Gehrig's, which causes other forms of weakness or muscle wasting. However, as I emphasized before, not all dementia is due to these neurodegenerative conditions. And as evidenced by, say, Parkinson's disease or motor neuron disease, though both can be associated with the dementia, that's not necessarily the case. So this is one way that people have traditionally thought of a progression towards a dementia. We start with just normal aging, and then frequently we encounter something called subjective cognitive concerns, where hey, things seem like they're tougher than usual, but if I get tested, I still seem to do okay on cognitive testing. And I'm still able to go about my life. Sometimes this progresses, though, to mild cognitive or behavioral impairment. In this case, not only are you or sometimes others seeing these cognitive changes, but we can also detect them on tests. At this stage, however, people can still independently go about their business. They can still manage their finances, they can still shop, take care of themselves. Maybe they need to write things down more or use other techniques. Maybe they're more frustrated, but they can still get by. Sometimes this goes on to progress to a dementia, which we've defined before, where you're having cognitive problems, they're detectable, and it is impairing your independence. Now, despite this strong arrow going from left to right here, I need to emphasize this is not necessarily the case. So sometimes mild cognitive or behavioral impairment or subjective cognitive concerns just stay there. And sometimes they may be reversible. Say somebody is just not getting enough sleep, or let's say that they're having a mood condition. Well, these are treatable. And once again, it's one of the reasons why we encourage early diagnosis and treatment. So I'm not going to belabor this too much because I know Dr. Potter is going to discuss it as well. But when we talk about neurodegeneration, you can see on the far left there a normal brain, and the Alzheimer's brain is more shriveled. It's shrunken because the brain tissue has started to waste. We also have a picture there of plaques and tangles. This is an example of what we would call proteinopathy, or a problem with the proteins where they become shaped differently than we see in healthy aging. This becomes a hallmark when we look at the brain under a microscope. We see different patterns of these proteins that are associated with different neurodegenerative conditions. So in Alzheimer's, we see these plaques and tangles. In Lewy body, well, we see Lewy bodies, little red dots. In frontotemporal dementias, we see still other complex misfolded proteins. This is a slide that is now just omnipresent in these types of conversations because it illustrates that these proteins probably start changing decades before people ever start having symptoms. So I hope you can see my cursor here. Here in green, you can see where people start changing, where we start seeing some of the signs of these brain changes. So over here, for example, you can see mild cognitive impairment, and that can go on, as we discussed, to a dementia. But well before that, you can start seeing, and this is based on genetic studies where we know people will eventually get Alzheimer's, we see changes in the amyloid protein. Again, many years before we ever see symptoms. We start seeing changes in another protein called tau. And then we start seeing changes on MRI where the brain starts shrinking. And all of this has really gone on for some time before we start seeing these symptoms. So I've talked about some of the benefits of detecting and diagnosing these concerns early on. Maybe something's reversible. But even if it's not, there may still be good reason to talk about this. One is there may be further lifestyle changes that could be implemented to help slow the condition down. Furthermore, there are emerging therapies that, if they're going to be effective, are probably best used earlier than later in the disease. It also helps make sure that patients and their family members are adequately prepared. It's important to plan ahead. Now, one of the newer medications that has come forward has caused a bit of a stir, certainly in our field. For a long time, the only medications that we had to offer boosted certain brain chemicals that helped us stay a little sharper a little longer. So these are medications like donepezil, also called Aricept. It boosts a chemical called acetylcholine in the brain that helps people stay sharper on the average a little bit longer, but it's not changing the underlying disease. Over the last few years, we've started to see what we call biological therapeutics, antibodies that target the protein amyloid and very convincingly remove the amyloid from the brain. Less convincingly and more controversially, it has some clinical benefit. You can see here on this graph, this is not stopping the disease progression. It's certainly not reversing it or causing the curve to go the other way. But we are seeing some difference from the placebo group, where people are just getting an inactive substance. However, with this has come some higher risk for AEs, that's adverse events, which lead people to stop it. Most of the time, these are fairly benign. However, sometimes they can be quite serious, including heart problems like atrial fibrillation, brain swelling, seizure, and other things. Nevertheless, because on the whole, it was decided that this is more beneficial than not, the FDA did approve this in 2023. It's still not very widely used, largely due to the extravagant cost of these medications, which run over $25,000 a year for this small benefit with these risks. And we are all still waiting to see what happens if it is more widely covered by insurance. So I want to talk a little bit about normal aging. So if you have walked into a room and you pause and you go, why am I here? What did I enter this room for? That happens to everybody. Same does that occasional tip of the tongue phenomenon where you're like, I can't quite remember the word or the name, a little bit slower coming up with things, harder to do more than one thing at a time. Again, this is something that happens to us all, unfortunately, as we get older. That said, as I promised, I want to stay focused on positives where I can find it. Some things just keep getting better as a rule of thumb. Vocabulary, wisdom, and kind of your general knowledge, for example. Where we start getting more concerned is when people forget what happened an hour ago or a couple days ago. If they're repeating conversations even when they were clearly paying attention at the time. We also worry about loss of interest in previously enjoyed activities, difficulty managing everyday tasks, and especially if this is concerning to others as well. Now, again, this is not necessarily the sign of something neurodegenerative or irreversible, but it's concerning enough that we would recommend an evaluation. We can also see things that go beyond memory. So while Alzheimer's disease is classically associated with memory changes, there are forms that can cause just problems with language or words. That's one of the things I primarily focus on. There are things that cause just disorientation. I don't know where and when I am, or just impair your judgment so you make bad decisions, or it can just present with changes in mood or your personality. So when we first evaluate somebody with these kinds of concerns, you can expect to be asked a lot of questions. That's because the brain is complicated and it takes a lot of questions to try and figure out what's going on. In addition, we'll do a neurological examination, bang on your knees with hammers, things like that, and a neurobehavioral status examination where we'll do at least a basic test of memory and other aspects of thinking. We also should and would recommend some picture of the brain, usually an MRI scan, and blood tests like a vitamin B12 and a thyroid level, just to make sure that it's not something that's more common and more easily stopped or reversed. Depending on the situation, we may look further with neuropsychological testing, lumbar puncture, more blood tests, or special imaging. There's no end to what can be ordered, but at the very least, we want to get the basics. Even in that sort of setting where, unfortunately, we do think that this is a dementia, there's still a lot that we can do, even if there's no cure. So, for example, we can still help by finding additional resources to keep people as independent as possible for as long as possible. We can identify sources of support, both in someone's own social or family circles and outside of that. We need to think about legal, financial, health decisions. We need to think about safety, driving, firearms. Not always right away, of course, but we need to make plans and engage somebody in the conversation. One more reason, and you're getting a theme here, why it's important to do this earlier rather than later, is that makes it more likely that the person with memory or thinking changes is able to meaningfully participate in those conversations and say what they would prefer. I also want to emphasize that caregivers or family members who are helping somebody through this process have to take good care of themselves as well. There's going to be a lot of challenges here. Relationships change. How you talk to each other can change. There are skill sets that need to be learned, and it's important to focus on staying healthy yourself as well. It takes time to get these sorts of things together. Coming back a little bit to some of the medical treatments that we can offer, we talked about the newer medications like lecanemab. We talked a little bit about meds that can boost brain chemicals to keep people a little sharper a little longer, things like donepezil, galantamine, rivastigmine, and memantine. Other things that we very frequently do is help control people's mood and help them sleep, because no one thinks well if you're feeling depressed, anxious, or just exhausted. So just honing in on a few things. Alzheimer's disease itself, wide spectrum, can present a lot of different ways. Every individual who goes through it is going on a different journey. It's not like your ability to do everything just stops right away. And a lot of what is widely considered Alzheimer's and what can you do turns out not to be. And so make sure that you're getting adequately checked. Now, earlier I talked about some things that might reduce somebody's odds of getting Alzheimer's. Of course, age is the big risk factor, genetics, gender, race, ethnicity. Those are things that, well, we can't really do much about. However, all of these things across our life are things that we have some control over. Amount of education, hearing loss turns out to be a increasingly prominent one. traumatic brain injuries, blood pressure, obesity, alcohol, exercise, diabetes, smoking, depression. You can see here that there actually is a fair amount that people can do to increase their odds of healthy brain aging. And with that, I'm just going to say thank you all very much. I'm going to go ahead and stop that screen share, and we'll move on to some questions.
So Peter, we're going to wait for questions at the end.
Oh, perfect then.
So we'll move right on to Dr. Potter's talk.
All right. Thank you, Dr. Potter.
Okay, thank you very much, everybody, for coming and joining us in this evening educational smorgasbord about neurodegenerative disease. And I want to leave you with some hopeful results at the end, but say that a lot more research is being carried out here at CU Anschutz and across the world. And we're not there yet with a magic bullet to reverse Alzheimer's disease. But as Peter indicated, there have been a number of really promising results that indicate that we're at least partly on the right track. So I want to emphasize just in the title that we need people in order to study a disease in people. But we also need mice, and you'll see how that's important as we go through, because together studying people and mice allows us to get insights into the mechanism of Alzheimer's disease, and then that may lead us to actual treatments. Okay, so who is Alzheimer? Alois Alzheimer in 1907 first reported a person who had clear pre-senile dementia. She was in her 50s and was as confused and lost as she said herself, with serious memory problems that might be expected of somebody who was 85 or so. And I use this as an example because, as Peter indicated, we're all getting older. And even though our individual risk of developing Alzheimer's disease may be declining because we're eating better and getting more exercise, generally living a healthier life, the definition of Alzheimer's disease has changed greatly since Alzheimer described this first patient. And that's because what the geneticists and the biochemists and the clinicians did in the 1960s and 70s is get together and say, look, what we used to call Alzheimer's disease, or pre-senile dementia, that is earlier than old age, is really the same disease as we're seeing in many people who are elderly. And therefore, we should call it all Alzheimer's disease. And the characteristic in the brain, as Peter indicated, is these plaques and tangles, which indicate a damage to the brain of August Dieter, the first patient, and of patients now with Alzheimer's disease. So these two pathological features that represent the hallmark of Alzheimer's disease has given us great insights into what's actually going wrong in both pre-senile dementia, in August Dieter's case, and in the classic age-related Alzheimer's disease that affects so many people. So here's what we're facing in terms of Alzheimer's disease currently worldwide. In the United States alone, there are about 6 million people with Alzheimer's disease. The annual cost is about $350 billion, and that's expected to go up to about $1 trillion a year for Medicare and Medicaid alone. That we absolutely cannot afford, either emotionally or financially. And of course, as Peter said, 10% of people over 65 have some features of Alzheimer's, probably 35% of people over 85. Women account for about 2/3 of the dementia patients and about 2/3 of the caregivers. So it impacts different parts of the population differently, and people of color are at greater risk both being a caregiver and of developing Alzheimer's disease. So that's the problem. And of course, Peter and I, being researchers, he's also a clinician, can tell you that what we need is more research in order to get to the bottom of what happens in the brains of people with Alzheimer's disease. Now, this is one of my favorite slides because it not only shows the young people and less than young people working in the laboratory, but it also shows on the left-hand side this person who you can recognize having Down syndrome. And people with Down syndrome all develop the plaques and tangles of Alzheimer's disease in their 30s. And most of them are demented by the time they're 50 or 60. And that's terrible for them and for their families. But it has given us great insights into Alzheimer's disease, because it turns out that these people with 3 copies of chromosome number 21 instead of 2 copies like everybody else has an increased risk of Alzheimer's disease, because the major Alzheimer gene is on chromosome 21. And that codes for the amyloid protein. So these poor individuals have a wonderful life if they can, but they make amyloid in greater amounts, and they begin to develop those plaques and ultimately the tangles in their brains much earlier. So here are the plaques and tangles I indicated before. The amyloid deposits are primarily the A beta, that stands for Alzheimer beta peptide, which is the component of the amyloid right here. And these plaques occur sort of like Brillo pads by the millions in the brains of people with Alzheimer's disease later in life, or younger if they have Down syndrome. Then the tangles occur inside neurons. These are outside of neurons, and they begin to kill those neurons, and they're made up of a different protein. And the combination of this pathology leads to the loss of neurons, the shrinking of the brain, as Dr. Pressman indicated, and also the loss of synapses. Those are the connections between nerve cells. You could have all the nerve cells in the world, but if they don't talk to each other, that's no good. And we won't get into it in great detail, but the brain of people with Alzheimer's disease also have inflammation, which is the non-neuronal cells, the astrocytes and the microglia get activated. Here's a picture of that. And Alzheimer himself noticed this back in 1907 as something odd in his first patient. Okay, so this is a classic figure that Peter Pressman also showed, in which the amyloid deposits start and rise to almost the maximum level before there's any clinical symptoms. The tangles come up a little later and the neurons begin to die. Alzheimer's disease may show up in the aged, but it is not a disease of aging. It's a disease that starts earlier and could be even considered a middle-aged disease. That means we really got to get a handle on it very early in order to be able to stop it. And one of the most obvious things to try to do is get rid of these plaques, get rid of this amyloid deposits. It obviously comes up early and it's part of the disease, so maybe we should try to get rid of it. So number of people around the world tried to develop ways to get rid of the amyloid deposits. And of course, in order to do that, you've got to be able to test your future drugs for Alzheimer's disease in an animal model. You can't just sort of say willy-nilly, well, we think we got this drug, let's try it in people. We need mice. And we could use other animals that get Alzheimer's disease, plaques and tangles. And obviously, monkeys are the closest relative of ours, and they do get plaques and tangles, but it's really unethical to use monkeys for research in large numbers. The other group of animals that we know develop plaques and tangles are polar bears. And Dr. Lillehei and the dean have said they don't want a lot of polar bears running around the medical school. So everywhere in the world, we needed a better model. And the problem with the usual models like mice and rats is they don't get Alzheimer's disease normally. But remember that I said that people with Down syndrome get Alzheimer's because they have a extra copy of that Alzheimer gene on chromosome 21. Well, it turns out there are some very special families who get inherited Alzheimer's disease. So if you're a member of one of these families and your father or your mother gets Alzheimer's disease, you got a 50-50 chance of getting it yourself, sometimes in their 50s, like August Dieter, or earlier. And that is because of a mutation, a change in the gene that codes for that Alzheimer gene, Alzheimer protein that's on chromosome 21. So all that people did was to take this mutant human Alzheimer gene out of one of these families, a little bit of blood, they can get the gene and they can put it into a mouse. And now this mouse gets amyloid deposits. And we can test that mouse's cognition by putting it in this little water maze. Mice swim very well, but they like to get out of the water. And this is just a kid's swimming pool with some channels in it. And at this channel, we put a little glass platform. It's underneath the water, so the mouse can't find it unless it bumps into it and gets out of the water. Now, normal mice, if you put them on this platform, they look around the room and they see where the door is, where the picture of Mickey Mouse is, or where the investigator is standing, and they memorize their surroundings. And then every time you put them anywhere in this maze, they immediately swim to the platform and get out of the water. And I promise you, we never let them drown. We always rescue them after a minute and put them on the platform so they can see where they can escape. The reason it's so important is that these mice are incredibly valuable. It costs more on a gram for gram or ounce for ounce basis to keep a mouse in our vivarium for a year than it does to pay a full professor's salary. So, of course, the professors are big and the mice are little, but we need thousands of mice, and we really appreciate them. So those of you who love mice, we love mice too, and they're very important for this research, because once you have a mouse that gets amyloid and has cognitive problems, then you can start trying drugs. And some of the first drugs that were used were designed to get rid of the amyloid. And so they then led to FDA approval of some of these drugs that are designed to reach into the brain, stick to the amyloid deposits, and teach the body to get rid of them. And that's what Dr. Pressman told you about in his lecture. Aducanumab was the first one. Lecanemab is most recently approved. And they actually are just a protein that's been made specially to go into the brain and get rid of amyloid. It works fabulously well. Here's an example of aducanumab removing amyloid. Here's where the red indicates how much amyloid a person starts with if they have Alzheimer's disease. And if they get nothing but salt water, 1 year later, they still got lots of amyloid, in fact, more than they had to begin with. But if they get aducanumab, a certain dosing, a larger dosing, and even larger dosing, get rid of almost all the amyloid in the brains of these people. A real breakthrough. The problem is, as Dr. Pressman indicated, is that there are side effects. Brain swelling and microhemorrhages, bleeding into the brain, occur in a surprisingly large number of the people. At the highest dose, it's about 40% of the people who are on this drug. The drugs are getting better. The problems are going a little bit less, but it's not a magic bullet for getting rid of amyloid. And the problem is that, as Dr. Pressman indicated in one of his slides, these drugs do not reverse Alzheimer's disease. They slow it. That's good, but it's not good enough. And that's where we come in the laboratory to try to develop new drugs. So one of the things that we've done is to ask the interesting question, which is there anyone around who doesn't get Alzheimer's disease? Well, it turns out that a number of laboratories, not ours, found that people with rheumatoid arthritis tend not to get Alzheimer's disease. And although it was reasonable, it's not because they're taking all these anti-inflammatory drugs, and we knew that inflammation was important, but that's not what curing their Alzheimer's disease. because a big clinical trial of these types of drugs in Alzheimer's disease was a total failure. So we went looking, and with a lot of work, which I won't get into, we found that a protein that is naturally in the blood of everybody and is increased in people with rheumatoid arthritis, called GM-CSF, which we won't get into the name of. It basically stimulates the bone marrow to make little Pac-Men. And these Pac-Men, these cells, go around the body and they eat up things that aren't supposed to be there. And it turns out that they're very good at teaching cells to get rid of amyloid in the brain. So here's an injection of GM-CSF in the brain of one of our mice. And this is just a control, artificial cerebral spinal fluid or saline. And one injection, 1 week later, got rid of 40% of the amyloid in the brain. So that was very exciting. And we could then also ask whether if you injected it under the skin, could the mice get better in their cognition? And it turns out, yes, they can. Here are the mice making errors in this water maze. Then if we treat them with GM-CSF, they're even better than a normal mouse. So they get rid of amyloid and they get cognitively better. Luckily, GM-CSF is an FDA-approved drug for other purposes. And therefore, we could start a clinical trial. And our first one, short and a small number of people, was positive when we published it. And here's the result. This is a measure of the Minty Mental State Exam. Whoops. That is a way of measuring memory. And the people who were given the recombinant human GM-CSF, called sargramostim, at the end of the treatment period, 3 weeks later, they were better than they started with, and they were better than the placebo getting salt water. And that improvement in the mini mental state exam actually continued for 45 days after they stopped taking the drug. So that's the first time that a drug has actually improved people's cognition, and was exciting. And the other thing that's interesting is that that same drug, GM-CSF, improved cognition in mice with Down syndrome. And even ordinary old mice like Dr. Lillehei and I are a little older. Probably our memory is great, but GM-CSF, if we were an old mouse, would improve that memory. Okay, so how does it do that? What we've just discovered, and this is unpublished, is that that GM-CSF seems to halt the neurodegeneration in the brain. We can measure the amount of nerve cell death by a blood test, and as people get older, even earlier and earlier in the younger ages, and especially in the old age, they lose nerve cells, and we can measure that in the blood. And if they take GM-CSF, that nerve cell death seems to go away. It drops it to the equivalent of a 40-year-old or younger. So that's good. And we're now in a clinical trial. Dr. Pressman is the PI, I'm the co-PI, or vice versa. And what we're doing is testing whether this drug can help people with Alzheimer's disease in the long term. So it's a 6-month trial. We got a $7.5 million grant from the NIH to do that, 5 GM-CSF injections a week for 24 weeks, and we'll see how they're doing. We won't know until it's finished. The fact that we could get a benefit to people with mice with Down syndrome has also resulted in a $4.5 million NIH grant to Dr. Pressman and myself to try it in people with Down syndrome. Again, we won't know the results for a number of years, but this is very exciting to bring these projects to CU Anschutz. I should also say that research is expensive. First idea about using GM-CSF for Alzheimer's disease came in 2007. The paper was published in 2010. We moved the team to CU and started our clinical trial, but it didn't get published and finished until 2021. And we needed funding in order to do all that preliminary work. We are very appreciative of the university, of the Global Down Syndrome Foundation, of individual donors, and even of the state of Florida and Colorado who helped us. And the conclusion I want to leave you with is the investment in science pays off. Now, are we resting on our laurels? Are we putting all our eggs in one basket? No, we have other approaches to developing Alzheimer's disease drugs. We want to try to prevent the formation of this amyloid. And here's one of the ways we can do it. ApoE is an essential part of the pathological process in Alzheimer's disease. You can make all the A beta peptide in a mouse brain or a human brain. By itself, it's harmless. It has to make that amyloid, and ApoE helps. So here's an example of our mouse again. If we get rid of the ApoE gene, all the A beta peptide is the same, but there's no amyloid. This mouse is cognitively normal. Okay, we can't get rid of ApoE in the human because it's an important protein, but we can see if we can get a drug that would block the effect of ApoE4. So we put A beta in a test tube, we allow it to make amyloid in the presence of ApoE4, and now we use drugs from the NIH, all the human drugs that have ever been tested, we test them one at a time, and we've gotten a few that actually block this effect. And that's very exciting. And since they're FDA approved, we can start a clinical trial as soon as we get some more money. Here's CU in the middle of the United States. All these blue states have Alzheimer's centers funded by the NIH. We're applying to get one here. And with Dr. Pressman and all of us at CU, we're hopefully going to get that this year. And I want to thank all the people that are on our team, because without them, and especially the research participants and their families, the philanthropists and the funding agencies, UC Health, we couldn't do any of this work. And I thank you. Sorry, I went a little over, but I get excited and I want you to be excited also. To be a little chauvinistic, I think the cure for Alzheimer's is going to be done right here at CU Anschutz. Thanks very much. And I'll stop sharing and open up Tammy Lack to look at the chat and see if we have any questions. And please do ask questions. We love questions. And if you can't remember them, then write them down at 3 in the morning when you wake up. Send Tammy an email and we'll get to you.
I'll get right on it if they do that. Okay, there's a lot of questions, so I'm just kind of picking the ones I can get to. Are there medications like lipids or long-term use medications for chronic illnesses that have been shown to increase the chance of dementia? For instance, is there evidence that statins taken for potentially high cholesterol preemptively block the enzymes that the brain needs?
You want me to fill that one?
Yes, please.
That's fine. So it's a complicated question, first of all. There are medications out there, such as statins, that have been associated with some increased risk of memory changes. Now, on the whole, physicians such as myself will still encourage appropriate use of those medications because the probabilities, again, I'm turning to statins here, are pretty low. So set as around 1% of memory changes in that class versus the odds of vascular disease due to high cholesterol causing more substantial and lasting brain injury. So we try to weigh those risks and benefits when we make our recommendations. There are other medications where we are more assertive in saying, no, you should really not be taking these. Those are medications, for example, that lower acetylcholine in the brain. So acetylcholine is the brain chemical that we boost with medications such as donepezil, but is lowered by even over-the-counter, easy-to-get medications like Benadryl, for example. which can really exacerbate cognitive changes, especially if there's underlying Alzheimer's disease and especially Lewy body pathology.
We have some very smart people on this Zoom, and the next question is, what are you doing with inhibiting the expression of unedited GLU-A2Q while maintaining expression of GLU-A2R?
Hunt, you got that?
Nope. I have to say that this is an area that I'm not very familiar with, but I do know that there have been many attempts to change the physiology of the brain in the mice in various ways. And unfortunately, many of these approaches have not led to a drug that can be applied in humans. So changing GLU is one thing that's been suggested, but I haven't seen data that I would jump to a clinical trial with.
A little less technical question is, why do you think more women get Alzheimer's than men?
That's a beautiful question. I think it's a very important question. Some of my colleagues are exploring it in great depth. There's a number of possible answers to that. I'll keep talking since I had to express my enthusiasm for the question. One is related to inflammation. So there are different inflammatory genes on, say, the X chromosome, of which women have 2. And that may alter the milieu in which Alzheimer's starts to take seed. There's a number of other social, environmental things that we could talk about, but rather than just hand waving, I'll hand the microphone to Hunt.
It is a great question, and one that we are working on in many ways. One thing that we've just discovered is that the loss of nerve cells in the brain that can be detected in the blood rises faster and rises further in women than in men. Now, does this mean that they stay dead and are not replaced? We don't know that, but we do know that the women seem to lose nerve cells in the brain faster than men. But, maybe they have more to begin with. Maybe they're smarter nerve cells. We got a lot more work to do before we can understand.
Okay, thank you. Do you check for Lyme disease and co-infections? This person had a neuropsych testing that showed cognitive impairment from Lyme disease or a co-infection, and then he or she had major improvement on IV antibiotics. Yeah.
So it depends on the situation. So here in Colorado, Lyme disease is not as endemic as it is, say, in the Northeast. In those regions, or for people that have that history of living in places where that exposure is more likely, then we are more likely to thoroughly assess. Now, I'm emphasizing this because there are also less approved labs out there where practically anybody who gets tested for Lyme will be found to have Lyme and then subjected to treatments. That I'm less enthusiastic about. However, chronic Lyme as a cause of cognitive problems absolutely can and does exist. I just want to make sure that the testing and the treatment are done appropriately.
Okay. This is a good one. Can you see the amyloid deposits or tangles on any kind of imaging while people are still alive, or only at autopsy?
Well, that was one of my slides. Those pictures of red in the brains before and after treatment with lecanemab was with a scan of the brain in a living person. So absolutely, we're able to measure amyloid in the brain and tangles in the brain of living individuals. But it's not something you can go to your local, PCP and ask for. It's a research program and is not yet used to diagnose Alzheimer's.
So there are situations where we will look for that amyloid clinically. Unfortunately, Alzheimer's is so common that it's not usually necessary. If you're over the age of 65 and are having gradual memory loss and we've done the basic tests, that's usually sufficient. However, if you have an unusual sort of situation, or if it's coming on at a young age, or there's something else going on that might be causing the problem and it would impact our treatment. And increasingly in the setting of these newer biological therapeutics where there's higher risk, higher expense, and we really want to make sure that we know what we're aiming at, there are additional ways of looking. We routinely get an MRI that's not possible to show the tangles, but it's to exclude other things. If we want to look for these proteins, we can do testing on cerebrospinal fluid using a lumbar puncture as well. It's very safe, but it is a needle in the back. You don't see people just getting it done for fun. There are blood tests that are coming forward. Most of us don't feel like they're quite ready yet for prime time and clinical use, but I wouldn't be at all surprised if we see them there in the next couple of years.
Okay, I think we have time for maybe one or two more. Are tangles and plaques in the brain also present in other neurodegenerative diseases like ALS or Huntington's?
Peter, you want me to do this one? Then you can talk about the difference in the clinical manifestations. The answer is, if you have plaques and tangles, you have Alzheimer's disease. If you have plaques and no tangles, you probably have pre-Alzheimer's disease. If you have tangles and no plaques, you probably have a form of frontal temporal dementia, which Peter knows best about, and I'll leave that to him.
Yeah, so thank you for that, Hunt. It does get very complicated in that there's actually a number of different types of tau tangles that can cause frontotemporal dementia. And there's another protein called TDP43, and there's multiple types of that. And then there's Lewy bodies. So there's a lot of different proteins that we can point to that are associated with these different conditions. For the most part, we don't look at those proteins, as somebody alluded to earlier, until autopsy. However, increasingly we are focusing on building these new technologies like the tests that we were discussing, PET scans, CSF, blood tests, that will detect these other proteins as well.
I have one more question that's a quick one. I think it's for Hunt. Hunt, you mentioned monkeys. Do we know of any other mammal that does get Alzheimer's or some sort of dementia?
Well, I mentioned monkeys because they're closest to humans. There have been some data in dogs and in polar bears. But in general, we need an animal model that's relatively small and relatively inexpensive. And so we're very, very pleased that we have rats and mice that have been made to get Alzheimer's or frontal temporal dementia or Huntington's disease by genetically manipulating them.
Got it. Thank you.
Well,
And-
we've
yeah.
kind of reached, I'm sorry, Peter, go ahead.
Oh, not at all. I was just going to say, unfortunately, I live with an example of another animal that does indicate some degree of cognitive dysfunction. It's called canine cognitive dysfunction. You can start seeing cognitive changes in dogs. And why that is still an open question, but some of these proteins that we've been discussed may be involved there as well.
Okay.
Right. Well, to respect everybody's time, we're a little over 8 o'clock. I want to thank Dr. Potter, Dr. Pressman. This was an exciting hour. And I certainly learned a lot, that's for sure. Again, anybody to reach out to Tammy if you want recordings of the previous lectures. We'll have a recording coming out of this lecture as well. And then any questions that we didn't answer, we're going to get those to you sometime later in the week. So thank you, everybody. Next week, we have a introduction to movement disorder and neuroengineering. This is a little bit of the Elon Musk Neuralink sort of connection. So that'll be exciting as well. So thank you all again for coming.